Good day everyone. Good day ma'am. So our topic for today is all about presumptive test and confirmatory test for blood and semen. So first, let me introduce myself. So I am Alex Bidul Santos from 3B Bachelor of Science in Criminology. So in this video, we have three objectives. So first objective, describe the blood and semen, the origin, composition, and nature. Then the second objective, describe the presumptive test and confirmatory test for blood and semen. Then the third objectives is describe the difference between presumptive test and confirmatory test. So, semen. Semen, also known as seminal fluid, is an organic fluid created to contain spermatozoa. It is secreted by the gonads and other sexual organs of male or hermaphroditic animals and can fertilize the female ovum. So, semen is produced and originates from the seminal vesicles, which is located in the pelvis. So, yun yung semen. Then the second one is blood. Blood is a bodily fluid in human and other animals that delivers necessary substance such as natures and oxygen to cells and transport metabolic waste products away from those same cells. In vertebrates, it is composed of blood cells suspended in blood plasma. So your blood is made up of liquid and solid, and the liquid part called plasma is made of water, salt, and protein. Over half of your blood is plasma. So yun yung blood. We have two categories of identification tests. The first one is the presumptive test or the preliminary test. It is the used for screening specimens that might contain the substance or materials of interest. So, the second one is the confirmatory test, a test which are entirely specific for the substance or materials for which is intended. And let's move out. The presumptive test and confirmatory test for the semen. So the presumptive test for semen, the first test is the acid phosphate test. So the acid phosphate test is a routine assay used to screen casework item for the possible presence of semen. So the color test in carried out of a filter paper, which is the retained after testing 2 years old test paper, were found to contain sufficient DNA for short tandem repeat profiling acid phosphate is present. A sample color will paper within a short period of time. It is almost 30 seconds. Weak stain may take between 1 to 3 minutes or 2. A possible reaction indicates the presumptive presence of seminal fluids. So as you can see in the image, this is the different features of seminal fluid. So the second test is the prostate specific antigen. It is also known as a P30, a glycoprotein produced by the prostatic gland and secreted into seminal plasma. Is a valid marker for detecting semen in evidence from criminal case, including samples deposited by chromized or azoospermic individuals. So as you can see in the image, There's a different test for seminal fluid. As you can see in the image, there's a different name in the mark. Yes? Yes. So we need to go to the confirmatory test for SEMEN. So the first test is the Christmas tree stain. So Christmas tree stain. Malapit na Christmas. A staining technique used to visualize sperm cells. So named because the different parts of the sperm cells take up different colors. Simplify microscopic identification. The tool shown as a yellow, green, and the head shape of red and pink. As you can see in the image, this is the test of Christmas tree stain. The head of a sperm cell is shades of red. The other is pink. So that is the Christmas tree stain. So the second is the RSID test for SEMEN. SEMEN is a lateral flow hemochromatographic strip test that used to anti-human hemoglobin monoclonal antibodies and lateral flow format to detect the presence of hemoglobin. Is a protein produced by the seminal vesicle and is responsible for the coagulum associated with the ejaculate. So this is the image of sample of RSID test for SEMEN. As you can see in the upper, it is the control line, in the lower is the test line. So that is the RSID test for SEMEN. The presumptive test for blood. So the first test is the phenolphthalein test or the Kastle-Meyer test. It is the blood, phenolphthalein and hydrogen peroxide. As you can see in the image, hemoglobin in blood causes the phenolphthalein to turn bright pink. As you can see the difference of the image, the one is positive, then the one is negative. So the substance and other blood may produce false positive result. That is the Kastle-Meyer test or phenolphthalein test. The second one is the luminol test. It is the chemical that exhibits chemiluminescence. As you can see in the image, striking blue glows when mixed with an approximate oxidizing agent. As you can see in the image, glow bright in the dark when it comes in contact with the blood. Diba? Grabe. So, we go in confirmatory test for blood. So, the first test is the Takayama test. When you say Takayama test, it is a confirmatory test used to detect blood spots. Based on the research result, this test can still be used to identify dried blood spots on clothing age 20 years. Grabe, di ba? And able to detect positively the presence of blood with the formation of pink crystal. So, as you can see in the image, this is the test. The blood spots with pink crystals. As you can see that. Yes, you can. Then the second test is the rapid stain identification test. It is designed for fast, easy, and reliable detection of human fluids from a variety of samples encountered by the forensic laboratories. So, it is useful for the forensic scientists. As you can see in the image, this is some tools or equipment used in rapid stain identification. Then the last confirmatory test is the ABA card hemate trace. So it is a testing device used to aid in the possible identification of human blood by detecting the presence of human hemoglobin. If human hemoglobin is present in the sample, its antigens will react with the mobile monoclonal anti-human or antibodies in the area marked. So this is the test of ABA card hemate trace. As you can see the difference of the negative, positive, and invalid test. This, as you can see in the image, as you can see this is the negative, then the positive and invalid. We go back to the objectives of this video. Kung na-reach ba natin? So, describe the blood and semen, origin, composition, and nature. Na-describe naman po. Describe the presumptive test and confirmatory test for blood and semen. Diba? We describe it. Then, the third one is describe the difference between the presumptive test and confirmatory test. So, na-describe po natin yun. So, that's it for today. So, I hope you learn something and you gain knowledge something from this video. Thank you for watching. Then may the Lord keep you safe and bless you always. Thank you. Shalom.